Hello, everyone, and welcome to today's webinar, From Insight to Impact, a conversation on the strategic role of biomarkers in drug discovery and clinical trials. I am Melanie of LabRoots, and I'll be your MC for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Olink Proteomics. To learn more, visit olink.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you are having any trouble seeing or hearing the presentation. I'd also like to now welcome our moderator for today's webinar. Chen is the Global Senior Director of Strategic Accounts at Olink Proteomics, part of Thermo Fisher Scientific. Ray, I will now pass this over to you.
Thank you so much, Melanie. And welcome to our panel discussion. Today we have an all-star panel of decades of experience and honor in the pharma and biopharma industry. And really what we want to accomplish today is, I think, threefold: to understand the increase in usage and strategic role of protein biomarkers specifically in drug discovery and clinical trials, and explore some of the real world applications of biomarkers in identification, drug profiling, clinical efficacy, safety, and mechanism of action, and learn how biomarker strategies can mitigate drug discovery development risk, drive better informed decisions, improve trial efficiency, and drive pipeline success in drug discovery and clinical trials. So before we get to all that and a lot of the questions that everybody have submitted online, plus some of the questions that we have for the panel that were planned, which is like the honorable panel here to introduce themselves. So Jay, I think you're the first one on my screen here to the left, so I will let you be the first one.
Thank you so much, Ray, and thank you for the opportunity. It's a true honor to, be here with my colleagues to discuss about, how we can leverage biomarker in terms of data generation and also interpretation and how that help decision making. So I'm Jen-Chieh Chuang. I'm a director for liver translational programs at Takeda, and before Takeda, I was leading many of the biomarker activity in the liver spaces at Gilead. And then collectively, I have about 10 years of clinical biomarker discovery and implementation experience. Looking forward to share some thought here today with the audience. Thank you.
Thanks, Jay. Dawn, I'll come to you next.
Thanks, Ray. So hi, everyone. My name is Dawn Waterworth. I'm at J&J Innovative Medicine, and I head up the computational group in Translational Sciences and Immunology. I'm really excited to be part of this conversation today. Proteomics has been something I've been excited about for quite a while. I'm so looking forward to the conversation.
Thank you, Dawn. So great to have you. And Magnus, last but not least.
Yes, thank you, Ray. So hi, everyone. My name is Magnus Althage. I'm based in Gothenburg, working for AstraZeneca, and I've been here in drug discovery and development in cardiovascular, metabolic, and renal diseases for about 20 years now. And the function I'm working in is called Translational Science and Clinical Development. And I'm, as my colleagues here, really excited to spend an hour with you.
Thank you so much to all of our panelists. So maybe let's start the discussion with the whole pharma drug discovery process, because I think, not everyone is very familiar with the entire process and how it works and all that goes into it, not just for money, but really, all the efforts and time these researchers go into developing the next generation drugs to cure all these diseases that we face, in today's world. I know when I first started in this industry, really did not understand, what really went in from the discovery side of things to development, to the clinical trials and the manufacturing. So maybe Dawn, I will go to you. to kind of explain a little bit of how, the drug discovery process works. As we all know, some of the numbers that we hear about, how many drugs, how many candidates really go into the pipeline and what the cost looks like. So from just your perspective and the traditional view, if we can briefly go through what that process looks like for our audience.
Oh, that's a big question. Okay, so I would say in terms of the process, the first thing we have to do is really understand our diseases that we want to treat. That's the first part of the puzzle. And then really identify, first of all, which diseases do we want to treat? Which ones where is there a big unmet medical need? And I think that's a question that we all think about as pharma. Once we've decided where we want to focus, then we want to identify our targets, right? And we can use genetics, we can use proteomics, we can use many different technologies to help us identify what those targets are. So once we identify that target, then the next step would be identifying some kind of drug or some way to modulate that target. That could be an antibody, it could be a small molecule. And then we start to try to optimize that. Once we optimize that, we get our candidate, and that's where we then do preclinical studies just to make sure it's safe. And then we go into humans. Typically first time in humans, you go in again thinking about safety, and then we start to gradually ratchet up into patients where we start to try and ascertain the dose. Once we know the dose that we want and everything looks good, then we'll move forward in clinical development, where we sort of go into gradually larger and larger trials and perhaps more precise patients. This whole process is a real team effort. There's literally thousands of people involved in this process. And I think I've heard on average it costs about 10 billion to discover and develop every drug. So it's really an immense effort, incredible process. But at the end of the day, you're kind of amazed that any drugs actually make it because it's such a long and involved process. But we are here where we are, and
Yeah, no, I absolutely. I think I'm fascinated every time somebody describes that whole process, from front to end, and really the effort that goes into it, right? So I just want to emphasize that a little bit. And Magnus, I think I've heard you describe that a few times, especially the part, where things start to go into humans from that preclinical to that clinical stage, and the trials of increased size, going from, toxicity measurements to efficacy, and sometimes, some of the biomarkers that has been used traditionally in this process, and the limitations that it had in the past, and and how that shaped the role of biomarker strategies in the past. Can you walk through us that bit a little bit?
Yes, no, I can do that. And I think traditionally, when I started here many years ago, we typically looked at biomarkers one at a time, and not much exploratory work was possible when you only looked at one. So the technology that has developed now into these multi-explore panels, where you can look at thousands of biomarkers in a very small volume, that has enabled us to explore the proteome, which was not there for us before. So I think we start to realize the value of this, and we know it's dynamic, the proteome, for good and bad, but we need to keep a handle on the variability, and we know there will be variations between individuals to individuals. But technology-wise, I think we move now, so we know that we can trust the data that comes out, and then we need to manage the other variables there. And I think, as you say, Ray, the process of developing a drug really, as Dawn described, we start small, build safety, build confidence, trust that the drug will do what we expect it to do in the right patient population. And it's like an upward funnel. We start with small, healthy, going into patients, and then maybe going into outcome trials with many, many thousands patients.
Yeah, that's right. And traditionally, I think there were a lot of challenges in implementing the biomarker strategy, especially on the proteomic side of things, with technology limitations, with how things were done from genomics to proteomics in the past. Jay, we've often talked a lot, with some of the work that you have done, specifically at Takeda, with some of the challenges historically that you have encountered, especially on the clinical trial side of things, right? And some of the things that you have done to address those issues. Can you maybe explain to our audience a little bit about that, just following Magnus's thoughts here?
Yeah, thank you, Ray. And just to echo what Tang and Magnus has mentioned, right? I think drug development is a Herculean effort throughout the process. There's a lot of critical decisions need to be made, and then to support go no go decision throughout the process, right? And I think the key here is people talk about data-driven decision making. And I would kind of agree only half on that, because data is a lot of 0 and 1. We actually need to be able to distill insight and actionable knowledge outside of the data, especially in the era of, we're able to generate simultaneously a lot of data point with evolving technology in multi-omic space, including proteomic. So I think the challenge, as you point out, Ray, right? So we also need to be evolved together in terms of not only being able to measure the analyte in a bigger scale, but how do we position the information and also come up with adequate plan, starting from sample collection and also with a predefined as much as possible in terms of depending on the context of use of the information, how does that support decision making, right? And then together with a very powerful computational or bioinformatic and statistician support in terms of filtering out the true signal and informative signal versus noise. So this is very superficial answer to your big question, but I hope this is gonna lead to a deeper and more extensive discussion later. Thank you.
Yeah, no. So Jay, maybe I'll stay with you a little bit on that. You've always have said, right, there's two really divergent uses of biomarkers, right? One is kind of understanding the disease, for better target selection, and the other is really to evaluate the treatment for faster and better treatment development, right? Can you, maybe extrapolate a little bit on that, just, to follow what you were saying?
Yeah, right. Absolutely, and I think so. I'm working on liver diseases, in fibrotic liver diseases, right? So there's so many ways that you can cause damage in liver. Fat, too much fat causing MASH, and then when you have too much bile acid, it leads to P.A.C.E. And then when you have AATD associated mutation, it causes this misfolded protein in the liver. So the point being, there's a lot of heterogeneity in terms of the patient characteristic, and then due to different etiology. So that definitely, represents some challenges when you think about target discovery and target identification, and also compound optimization, etc. Right? And then I think to your question, there's going to be all this key important process that you need to make a proper decision, evaluating the risk versus the reward that certain compound can bring to the patient. And then I think, at least from my end and based on my experience, the step one when you design and come up with a biomarker strategy, you always need to ask the question properly to begin with, right? Depending on to Dang's early point, what is the unmet need and whether it can be addressed by a biomarker, and what type of the data set that you want to generate to address this question. We need to remember that there's very complementary nature of different data set. In the clinical trial sample, you do have a very in-depth characterization of the patient, and then it also has treatment response. In a translational study, for example, you'll be paying more attention to the disease physiology versus healthy. And then in nature history population, data set, there's going to be a broader data, but not necessarily as deep. So I think, to begin with, you do need to define your question, identify the right sample and data source, and generate the data, and working with the data scientists to really filter out the useful information. And last but not least, I think we also do need to acknowledge the requirement for the data, and also the assay development really will be anchoring on, how do you position the biomarker. If we are talking about endpoint, secondary, and even primary, the performance, the analytical performance, and also the clinical validation need to be very robust for that particular context of use, right? So again, just to come back to your question, there's going to be a big scope of application, but really depends on the context of use in specific setting. We do need to have a customized data generation, data analysis, and analytical performance validation plan accordingly. Thank you.
Yeah. No, that's a great point, Jay. And I just want to remind the audience, there is a live question that a box there where you can put in questions, and we will answer them as the discussion flows, and not necessarily at the end. So please input these questions whenever you think of them. So that's a great point that you had there, Jay, in terms of data generation, what kind of data qualifies, and the quality of the data and what is useful. So maybe let me back up a little bit here. Before we really started these large scale proteomics projects and generation, traditionally we had genomics, which really dominated the landscape for about 20 some years, as a quick way to assess a lot of information. And we really, during the genomics revolution, we thought, that was the end all be all. We're going to solve all the diseases in that particular way, until we realized, hey, there's a gap, within that biological dogma where, things go from RNA, DNA to RNA to protein, and there's a lot of pathways that could take. And the proteome, which is the closest to the phenotype of everything, really is a key point of discovery. So now we have all these proteogenomic studies, which is the integration of genomics and proteomics. And Dawn, I know you have been really involved, in one of the largest, I think the largest proteomic studies today, which is the UKB Pharma Proteomics Project, incorporating, I think, 13 or 15 pharmas these days, where a project of a scale of about 600,000 patient samples going underway. Can you maybe talk a little bit about the driver, expanding on what I just had mentioned, of the thought process of that project, how it all initiated, and the rationale behind the pharma backing of it, and what do you hope to get out of a project like that?
Yeah, thanks, Ray. So I just want to underscore a point that you just made, which is a very important one. 20 years ago, when I was doing a lot of genomics, we thought genomics was going to give us all the answers, right? It's going to help us find the targets, and it's also going to help us stratify our patients. And that was the prevailing thought for a long time. But it really hasn't panned out. The genomics piece is really good for targets. Yes, that's a real strong value, and that's been well validated now. But in terms of patient stratification, it's really been a little bit of a bust. Good for rare diseases, yes, but not for common diseases. So if that's the case, we need something else. And as you just said, protein is closer to the disease, and in some cases, a very strong disease biomarker. So I think that was part of the mindset behind the UK Biobank Pharma Proteomics project. Can we take this data set, which is very rich clinical data and biomarker data and genetics, measure proteins here, and really explore that data to see what we could understand about the patients and the diseases that we're interested in. The other thing that it did was we need in pharma, we need a platform that's very scalable and consistent. This is really important. I mean, ideally, I want to take a platform and I want to go to my clinical trials, I want to go to my collaboration data sets, and I want to have the same data. I think in the past, sometimes we had one platform and one data set, and another platform and another data set, and maybe just a subset of patients. That's just not useful, right? Ideally, you want your data sets to be interchangeable, that's going to maximize the value of the data and how much you can extract from it. So I think UK Biobank, what it did is it really validated that Olink was a very scalable and consistent platform with high quality data. And that gave me the confidence to move forward thinking, okay, this is something I should be measuring in my trials and in other collaborations. So now I can start to build up a large data set that I can easily look across diseases and across studies. So that's critically important. The value of the data, I can trust that data and then use it to make decisions. Because we are using this data to make important decisions for patients. So it needs to be reliable. And if you have data that's not correct or has issues, that can cause you many problems. You can waste a lot of time as you move along.
Yeah, no, absolutely. And maybe let me stay with you for 1 second with all the proteomics data set that you've mentioned that has been generated. Obviously, Olink has become a really trusted technology in that space. And of course, there are other platforms out there, right, that we think are complementary at times. So, Cindy had here just asked the question from the audience, how do the speakers feel about integrating something like mass spec proteomics alongside affinity proteomics like Olink? Maybe I'll get your thoughts since you were on this subject.
Sure. So yeah, mass spec has a lot of value, and I think that platform has also been evolving a lot over the last few decades. I've been hearing more about it recently, and it's really made incredible advances. I think perhaps one of the values there is going to be in looking at tissue. I think when you measure proteomics in tissue, you get with mass spec, you get a lot more proteins. Right. So that's, I think, one of the places where it can be really useful. And I think it has a very strong place in discovery where you want to measure everything. Right. So I think these panel platforms are really good when you want to go in and sort of get very consistent data. And it's not, you're not, boiling the ocean here. You're getting a set of proteins that you might be interested in. So we've made the decision already what we're interested in. But in discovery, sometimes you want more, right? Because you just don't know what you don't know. So I think the place for LCMS is probably more in the discovery space, and it's probably also more in tissue.
Yeah, no, those are both good points. I think the matrix plays a really significant role in terms of the different technology you use. Some matrices are just more applicable for certain platforms. And then there's the whole discovery versus what goes into clinic, and what's targeted and what's more discovery. I think that's a great answer for that. And so maybe let's stay on that topic. Magnus, I'm going to come to you. This is a little bit of a challenging question that I've now gotten, I think, 2 different questions from the audience on this topic now, which is, there's a lot of different informatics tools these days, for proteomics data in terms of, how to utilize that data more effectively in, finding the biological meaning and specific biological states. And of course, AI is also playing a major role these days in terms of, bioinformatics and how to, be more efficiently, if you will, to utilize that data towards drug discovery and patient treatment. What are your thoughts on, utilizing AI and other bioinformatics tools in this space once we have this whole crunch of data? I know AZ is also part of the UKB project as well, right? Where a lot of data is about to be generated.
I do think that AI has a fantastic potential to help us here, because when we build on this wealth of data, being proteomics, being lipidomics, metabolomics, you name it, it's hard to manage to see patterns and to see the finer details. So there, I think AI has a great potential. And it's not the way I look at proteomics. It's not only looking forward in an ongoing trial or identify a patient. We know we can do that, but also the reverse translation. And I do think that using our later studies, maybe phase 3 studies, to build understanding how the drugs are working, with pathways are being regulated, there I think AI can help us to identify new targets, or maybe even new combination of drugs to better treat patients. So I definitely think there is a need when technology can generate more complex data we need to have analytical tools and power to harness and to maximize the use of the data. And their AI, I think will make a huge difference.
Yeah, no, absolutely. I think it's interesting you touched upon, phase 3, reverse translation, right? Finding out the mode of action of specific drugs and the pathways that it's affecting. There's also thoughts on, utilizing the proteomics data in terms of, having a drug fail faster. I think that's also a source of efficiency in the drug discovery world and processing. Can you mention a little bit of that and how the data has been utilized in terms of, maybe killing off a program earlier than you would have, and what are some of the challenges there?
Yeah, I think coming back to, so the way we tend to use it early is more building confidence that you hit your target of interest. And there it might be a handful of biomarkers that can show you that, or maybe even just one. And to identify that biomarker that is related to the target, that's very important. But the next step is when you then move into clinic, it starts to be more and more expensive. If you know with which pathways you want to regulate in a specific disease setting, you can address them early to see if you see signs of regulation. If you don't, or if you see opposite signs on pathways you don't want to act on, being maybe safety signals, you have the opportunity to detect that sooner than later.
Yeah. Jay, I see you nodding there. Do you have any additional thoughts on this?
Yeah, right. I totally agree with what Magnus has mentioned, right? And I think again that there's going to be a lot of key decision to make with a lot of string attached, right? So a quick kill is going to be a success already in many cases. And I think the principle, just from my perspective here, is you do need to, as soon as possible, as Magnus has mentioned, right, pick up the right target engagement biomarker. To educate yourself in a way that whether your proposed compound or RNA or antibody, regardless of what platform, can indeed engage the target of interest sufficiently. And then, of course, with acceptable and optimal safety profile, you can propose and support a goal decision to further test whether, when you have a sufficient target engagement, does this change the disease course or in the disease population? So in that setting, the early proof of concept study in disease population becomes super important. There you will be not only looking at sufficient target engagement, but you also want to investigate and come up with a suitable pathway modulation, disease modulation biomarker. And this is still in the early stage, so the N number is the key here, right? And to your question about how biomarker can help, if you don't define the quantitative criteria properly, you may have a huge early stage program, and then the decision making may not be effective, right? So you're wasting a lot of resources to come to a point of decision making. So the power of biomarker in this setting is once and assume that you can identify a suitable target engagement, pathway modulation, and disease modulation biomarker. They can actually serve to some extent a predictive biomarker for your late stage decision making. So in a way that you kind of de-risk early on with reasonable patient numbers before you commit it to a much longer and bigger scale, which rely on the endpoint for approval. So those are some considerations in a way, just add on what Magnus has mentioned.
Yeah, so Jay, maybe I'll stay with you here a little bit. You just mentioned some of the decision making that you are able to, do with these biomarkers and the answers that you're getting, from these trials. What are some of the regulatory concerns, associated with biomarker-driven approaches, right? Were there any, steps being taken to ensure that these approaches are within or aligned with the regulatory requirements, or is it you're at a step in this process where that's not a concern at this point? Can you extrapolate on that a little bit?
Yeah, thank you, Ray, and I'll be sharing my observations and perspective because my expertise is definitely not in regulatory space, right? But the one point that I would like to make is I think even the phase 3 primary endpoint or the pivotal endpoint, they were all biomarker at some point. So you're measuring, some measurable signal, and then affiliated that signal with clinical risk or clinical outcome, and then the precision of the measurement and also the link between the signal, particularly the changes of the signal to the outcome. That is somewhat critical in terms of being able to accept certain biomarker to be an endpoint, a provable endpoint, right? So to your point, I think, at least from my perspective, this is going to be a long process in terms of qualifying a biomarker to the level of endpoint. But even before that, I think throughout the different decision-making checkpoint, right? Biomarker has a lot of potential as, not at the level of regulatory just yet, but for decision making, I think it's going to be very powerful. And then there's going to be associated requirement of analytical performance, of course, right? As program advance to a more mature stage, the requirement is going to be higher and higher. I think that should be part of the consideration when we position the value of biomarker can bring, right? So it's going to be a big spectrum. Initially, you started with a big panel or platform like Olink to give you a lot of information, a lot of knowledge potentially, and then you nail down to a smaller panel that while you establish the link with those marker with disease of interest and potential risk, and then you go and continue to build evidence and refine the analytical performance, and then eventually maybe one or two of these biomarkers can become endpoint one day. And I'm saying this because in a way that in the liver fibrotic disease, the field is still facing a challenge in a way that you have to rely on biopsy as a provable endpoint in many liver diseases. And people are trying to walk away from this invasive and also notoriously heterogenic, endpoint, right? So if we can come up with something that is non-invasive, and then it can be reliably measured in the blood, with Olink and some of the markers potentially derived from Olink, and then further establish the link between all these non-invasive biomarkers to liver-related outcome, then it's going to be one step closer to, walking away from, the dependence on the invasive biopsy, right? I hope this addressed your question, right? Yeah.
Yeah, no. That's great. Yeah, so I think there's still some obviously regulatory challenges, but I think we're making our way there in terms of incorporating this strategy, which I think has made everything really more efficient and helped in this drug discovery process. So I think when we dreamed up of this panel a few months ago, our thought was, hey, there was a period of time when we didn't have this strategy incorporated in the process, right? And we found all these benefits and uses by using protein biomarkers, in this whole process. And Dawn, I think what we had talked about was, hey, were there any tangible benefits that you found by utilizing, Olink or just proteomics in general? There was a before and after, right? And what have you seen as some of the really tangible benefits, that got you to say, hey, let's utilize this in all of our, immunology kind of platforms and our pipeline?
Yeah, thanks, Ray. That's a really good question. I mean, in the translational space that I work, we need usually two types of biomarkers: one peripheral biomarkers and tissue biomarkers. And depending on the disease that you're working on, one can be more important than the other, but usually you would like to have both. If you have tissue biomarkers only and you don't have good peripheral biomarkers, then it makes them really hard to apply as you kind of move through the drug development kind of pipeline. Now, if I look back 5 years ago when I joined J&J, we really had very few peripheral biomarkers. We were mostly using RNA-seq, and even before that, RNA arrays. And we had a very small number of proteins. And what we found, and now that we have protein data and we look at our PBMC or blood RNA-seq data, it wasn't that good for generating peripheral biomarkers. I mean, there was a few successes here and there, but what we're seeing is that proteomics is just giving us a lot more in terms of identifying biomarkers that are really relevant to our processes. So I think that's been the big win. We now have much better distal biomarkers, much better patient response biomarkers, for example. And just to pull out a couple of examples that we've been working on, in IBD, most of our biomarkers were from tissue. And what we were seeing in RNA-seq from PBMC, we weren't really seeing much, right? We actually did a lot of sample processing, generated a lot of data. Very little came out of it. But with proteomics, we're starting to see now some peripheral biomarkers that we can correlate with those tissue biomarkers. So that's really valuable. And then also another example in psoriasis, we were interested in super responders to one of our drugs. And proteomics was actually quite a useful predictor, along with some clinical parameters in identifying those patients who would go on to respond really well to one of our drugs.
Yeah, and maybe a related question here from Laura. Is there an optimal stage, in the whole drug discovery process and development to begin exploring protein biomarkers as part of the strategy for a given asset or a series of compounds?
Who do you want to take that?
Yeah, I'm sticking with you, Don.
Okay. Okay. Is there an optimal stage? Well, I think you want to start from the beginning, to be honest, because as I said at the very beginning, you need to understand your disease. Right? So right back when you're doing discovery, the more you understand about your disease and disease biomarkers, the more they can then help you when you're in development. So they're kind of two sides of a coin. So I would start early, build up your knowledge base, build up your data. And then once you get to those tricky stages in development that we were just talking about, you're just more informed. So you're interested in response biomarkers to a particular drug. The more you understand about disease biomarkers, then okay, I can look at these response biomarkers that I've identified. Is that also a disease biomarker, or is this something specific to the drug? So you really need to understand as much as possible. And this is why it's really good to have sort of patient, sort of like disease control data sets, like usually from collaboration or from resources like UK Biobank and FinGen, to really understand our diseases there. And then when you go into clinical trials, it's very complementary. They're generally much smaller, but we've learned from these larger data sets where we've got different layers of data. And that then helps us use those protein biomarkers in the best way possible.
Yeah, no, that's a great answer. Thanks, Don, for sticking with you in that very specific question there. Yeah, so Magnus, maybe I'll come back to you now. You just went through the last time with the answer of looking at the trials and the expansion and what's your experience in terms of like how did you first get onto the protein biomarker train and the benefits, and what are some of the challenges that you have seen on the flip side of this?
Yeah, well, maybe there were two things. I think to in the beginning, everyone knew about genomics. We knew about DNA, RNA. That was kind of easy to justify. But then proteomics, it's so dynamic that I think in the beginning it was questionable how much value can you actually get from it. But I think by generating more and more data, like you described here in UK Biobank and more, we started to see really the value of it. And I think that has been a process to generate data showing the value. That is a key thing. And then also in I think really to Dawn's point, I think you described it really nicely there, when to start using proteomics like this. And I really agree with that. I think the earlier the better. And we have some occasions where it made sense. We already started to explore mode of actions preclinically, even going into human settings where it made sense. And it turned out to be very valuable. So that really building confidence, showing how your drug is working, that makes development much, much easier.
Yeah. Maybe, I'll stick with you now, Magnus. Is there, and you can answer this question very generally, just in your work that you currently do at AstraZeneca, are there any, examples where I know you sit in the translational space, where, early clinical and late clinical, you kind of talk to both sides of things, right, and make a decision based on some of the discoveries that you have made by utilizing protein biomarkers, or just the large tranche of data that you have seen from some of the population scale studies that you have seen.
Yeah, no, but that's true, and we do that a lot. We work cross-functionally, and I think also but it is different to look at a proteomics data set compared to just looking at one biomarker at a time. Because traditionally, it has been a lot about statistics, and it will be at the endpoints that we take go, no go decisions on, typically are statistically driven. But looking at the proteome and multiple biomarkers, it's not always statistically significant. It is trends. And I think that is something that takes some time to understand and live with that kind of uncertainty, if you see it that way. Yeah, so no, but for sure, it has been a challenge also going into later studies. Should we do a broad proteomics assessment in a phase 3? Do we risk finding something that we might not want to see? But I think really the balance is now that the benefits are stronger than the potential risks, because we can find so much more valuable things there.
Yeah, absolutely. I remember, even in the mass spec days that I was in, right, as the instruments and things get more and more sensitive, it becomes a question of like, oh, do we want to see all these things that we have not seen before? But I think, when now that we're seeing all these things, it definitely, I think, creates more value, and I think helps us solve more questions that we've had before, that we may not be able to answer, because the technology has advanced to this stage. Yeah, no, thanks Magnus for that. So I'm seeing a lot of questions now popping in from the audience. So I'm just going to, in the remaining 18 minutes we have here, kind of go through them in terms of some of the related topics that we've discussed. So Cindy has another question here. I think this is towards you, Jay. Cindy saying, hey, I wonder if there's a future where a biomarker signature specific to a disease, or maybe all-cause mortality, might be used as a surrogate endpoint to enable more efficient clinical trials. Yeah, so this might be an extension of your answer previously, Jay, if you want to talk a little bit more about that.
Right, absolutely. I think I totally agree. I can totally see the vision, right? Because again, currently the field is relying on a suboptimal tool, which is the histology in liver biopsy, right? And I think down the road, and it may not be just one marker, and again, with the appropriate multivariate analysis, you may be able to find a couple of very informative and important biomarker that can really clearly link to the clinical outcome. So in that regard, this non-invasive, it could be a combination of biomarkers coming from Olink, right? Together with some imaging tool. So I totally see the value and potential. However, this is going to take some time and some effort, going back to the clinical validation part, right? So this need to go through a very robust and careful validation, and getting the buy-ins from the regulatory parties and the support from them. I think we started to see directionally this is where the field is going, at least in the liver spaces. But we do have a lot of more work to do, right? And with that, I also want to make a slight point in a way, because we're talking about when to start to do this, right? And I totally agree with Don and Magnus in terms of the sooner the better. There's an element of, in a way, that when patient committed to clinical trial, as a translational biomarker scientist, I view this as a huge privilege in terms of being able to use the sample to generate information and then to benefit the patient down the road, not only for the drug development, but also understanding the disease, right? So there's element here in a way that there's only certain amounts of sample that you can collect in the clinical trial. So how do you maximize the value of those sample being collected? So in terms of being able to generate hypothesis or test your hypothesis. So per unit of sample that you have collected, if you can maximize the number of data points that you can acquire, then you're going to have a better chance to generate better knowledge, right? So in that regard, you want to do it early, but you also want to maximize the output of the certain, blood or tissue sample. I think that is an important consideration as well. Thank you, Ray.
Yeah, thanks, Jay. That's a very comprehensive answer. I think Cindy would be satisfied with that one. And hey, Dawn, I see a question for inflammation immune response. So Ugo here has asked, has there been a great progress in linking biomarker data to specific biological states, for example, inflammation or immune response? So in your experience, have you seen some of that come to fruition?
Yeah, I mean, I think it's still early days. We've just had a lot of this data come through in the last year or two. And I think there's a lot of excitement, a lot of promise, and we're starting to see some very early signs that this is going to be the case. But I would say it's still early. Immunology, has tended to do things in a very traditional way, using these cell surface biomarkers for many, many years. And that's kind of the more normal way to move forward. But I do see proteomics being very complementary to that. And I think ultimately it's has potential to be a much more straightforward biomarker to be used in the clinical space. So we're in the midst of that now. We have some early signs and a lot of promise, but more work to be done.
Yeah. No, I totally agreed. I think, as excited as we're getting, about all the strategies and data that's been generated, we're really at the forefront of this, I think next phase of drug discovery and proteomics revolution. So I think the more exciting things are, to come. And as to Jay's point, right, there is a lot of regulatory stuff that's kind of in the works at the moment. I think, some of it with Olink as the technology backbone backing some of these efforts as well. So, that's all great to hear. All right, so I'm just going through these and rotating amongst the three of you. So Magnus, this is a question that might be a little bit challenging, at least for me. So I'll test this on you. So this is from Sarah Gomez at the University of Guelph, and so she asked if extracellular vesicles hold a lot of promise as a source of novel protein biomarkers, but their inherent heterogeneity often complicates reproducibility and standardization. From your translational perspective, what qualities would EV-derived data need to demonstrate in order to be considered useful or actionable in drug discovery and clinical trials?
Yeah, so extracellular vesicles, they have potential for sure. Typically, there's a sensitivity challenge to be overcome. To start with, that you will be able to detect it in plasma or urine, for example. But then you have both you have the biological variability, and then you have intra-individual variability. And they can't be too much. They can't be too large, because then you can't really draw any conclusions. So finding an extracellular vesicle with all the challenges around that in terms of finding ways to isolate the extracellular vesicles in a consistent manner. But if you do that, then I think it's sensitivity and it is the biological variability that really make or break extracellular vesicles in a specific patient population.
Yeah, no, absolutely agreed. And just a side note, maybe a little bit of a advertisement here, but we are also finding that Olink is great tool for EV work. Recently, we're seeing a number of publications with EV content and our Explore HT platform as well. So yeah, I think that this could be a source. I think Magnus, you're right. There's some problems to overcome and challenges to overcome there in terms of sensitivity. That's for sure. All right, coming back to you, Don. In your view, what have been the most persistent misconception about biomarkers in drug discovery, and how have these perceptions shifted over time? I'm giving you all the historical questions.
I'm so old. Persistence, it's an interesting one. You mentioned specific discovery. That's what the person's interested in.
Correct.
I would say animal studies, the predictive value of animal studies, in predicting whether a drug's going to work or not. I think that was something that discovery perhaps didn't necessarily get right. It works in some instances, but in many instances it doesn't. You want to be focused on humans. So the more data you have from humans, the better, because that's going to be ultimately much better in terms of predictive context. And perhaps the other thing to mention here is proteomics also goes really nicely with genetics. Right? And using proteomics can be one of the ways that we do variant to gene mapping for genetics, and that can help us in understanding our genetic signals and our GWAS signals. So I think there's a real value there, and especially with the advent of pQTLs and helping us to localize some of our signals and helping us sort of head off in the right direction. And that's been a real leap forward. So I'd say using human data more, less animal model data, but also complementing genetics and proteomics together being really valuable in terms of prediction and charting a course forward into development.
Yeah, no, that's a great point, and maybe a follow up. Just my own thoughts here. I feel like when I talk to some of the KOLs nowadays at pharma, right? There's kind of like two schools of thoughts, right? One is like that historic, like, hey, we go from preclinical, right, mouse model, to humans and to all that, and then there's some other people who think, hey, mouse actually make for really terrible human models. Let's do these large population level studies to really find some of the significant data points for which we can have some really actionable data to go after these real biological functions. I know, Don, we mentioned the UKB project. I know you're also part of that 3TR, consortium project in Europe as well. So obviously, I feel like you're a believer of this large population, kind of cohort studies with data. How do you see these two different kind of views, in this process of drug development, and how do you reconcile them, or do you see, that hey, there's usages in both sides?
No, I think there's uses in both sides. I mean, animal models are still useful when you're trying to understand mechanism of action, for example. They're just not as predictive as we initially thought they were. Right. So I think with this sort of movement towards using more human data, what we've realized is we need a lot of it. Right? So this is why we have all these collaborations and all these large data sets. And for many years, we had all these like relatively small sample sizes, and we tortured it to death, and we find a few things, but we kind of realized over time, actually, we need more data. And now that we have more data, we can really understand which results are really solid and which ones are not. Sort of looking backwards, we realized that perhaps some of the data we had 10 years ago wasn't so good. So given that, we really need to build our knowledge base, and this is part of the reason why in J&J we're involved in many of these collaborative efforts, because that's part of how you do it. It's a way to access large data, also work with other experts in the field. And I think we're all trying to learn. We're all trying to learn about these diseases we're working on and which biomarkers are the most useful for the many functions that they provide. So I think we're in an exciting stage. It's massively taking off, it's expanding, and I think you're going to see a lot of progress in this space in the next few years.
Yeah, no, very excited. I think that this goes back to your point earlier, where we're at the very beginning of this revolution. So Magnus, I'll give you a historic question now. So like if you look back at all the years that you've been doing this, what do you feel like is the turning point for you in terms of like, hey, the way we're doing things traditionally have not been enough. Let's utilize this new, kind of strategy or technology.
Yeah, if I were to pick one thing, I think it's around the potential for precision medicine. I think we talked a lot about monitoring drug effects and mechanism and so on, and that's good. But to be able to identify a responder and not just go for all comers, which we traditionally did in cardiovascular diseases anyway. So now, on a molecular basis, dissect these heterogeneous, clinically defined patient populations. I think there we have a fantastic opportunity to go broad first, explore, turning it down to Jay's point. For validation reasons and regulatory reasons, you need to go down to maybe a handful of biomarkers that will identify your patients. And that's the patients you take into your studies.
Yeah, no, I think Jay, you could probably expand on this. I mean, I think that precision medicine point is really key here. That's what kind of our dream is and what we're driving towards, where, each patient, right, you're talking to, somebody like Jenny Van Eyck at Cedars-Sinai, she talks about your kind of clinical twin, right, where really, you're basing the treatment of this very specific patient on some of the data we have for someone in a very similar, genetic, proteomic, or disease progression makeup. And I think your small panel really kind of points towards that, right? If you want to just expand on the precision medicine point a little bit here.
Exactly, Ray. And then I totally agreed. Again, I've learned so much from my colleagues today. This is fantastic. So, and it's come down to a question of patient segmentation, right? Knowing that there's all this heterogeneity, and to Magnus's point, how do you put the different patients at a high level? Their symptom may be similar with various severity, right? But biologically and from the biomarker perspective, they may be very different, and that is actually going to make up to whether you're going to have a response to particular MOA or particular drug or not. And then that is definitely a stepstone that we need to overcome to have a precision medicine like approach, right? And I also want to, coming back to your question about the misperception, right, of biomarker. I think historically people use, usually you don't think about generating biomarker data unless you have a problem. So if everything goes well with primary and secondary endpoint, you don't need biomarker. And I think that has changed quite a lot. And also going back to the point that Magnus and also Dan has in earlier, the translation, reverse translation element is actually gonna become more and more important, right? We're gonna be able to leverage the data that generated in late stage or even earlier stage clinical program. It's gonna further educate us more about, again, the heterogeneity, mechanism of resistance, and then potential combination opportunities. So the reverse translation part is becoming more and more obvious and valuable compared to the historical concept about how people use a biomarker to solve problems. And this is really an exciting era. I think we are going to be have some fun down the road.
Yeah, absolutely. All right, we have 3 minutes left. So last question for panel, and I'll come to each of you real quickly. What's your bold prediction about the future role of biomarkers in clinical development? Are we moving towards a biomarker-driven drug pipeline as we stand today, or what's your bold prediction for the future here? Dawn, I'll let you go first.
Yeah, I think it's going to be in patient stratification, as was just discussed. I think proteomics has given us an opportunity to really help us sort of pick apart those different patient subsets that we know are there. And we just really haven't had the tools to do that. And then that's going to allow us to really target specific patients that are matched with our assets that are coming through more precisely. So I'm really excited by that, making a lot of progress. And I think that's going to be the biggest place where we're going to see progress.
Very good. Wow. And the lights went out on that. I mean, it was perfect.
Mic drop.
I know you planned that. Yeah, Magnus, I'll let you go second.
Yeah, so Dawn, I couldn't agree more, really. I think that's where we will see the successes in the near future. That is where we identify the patients to treat based on proteomic profiles and genomic profiles in combinations.
Yeah. Jay, leave you the last word.
Yeah, thank you, Ray. I think, I beyond what Magnus and Don had highlighted, right? I think there's also an opportunity that, we can seek to make drug development more effective in terms of the time and also the amount of investment. So we can really bring the drug to the suitable patient for treatment much earlier and, much economically, affordable, right? So I think in the long run, I think I do position, biomarkers, including Olink and the affiliated protein biomarkers, right? In terms of really giving us an opportunity to having a more effective decision-making and drug development process, so we can bring the drug to the patient quicker and, with better price.
Yeah, no, absolutely. I think that's really the dream for us here at the end. And I think proteomics is, with these really advancements in technology platforms we're on the process, right, of enabling a lot of this into the future. And I'm very, very excited about it. AI certainly is part of it. There's a lot of question about AI. I couldn't get to all of them, but really, we will try to answer them. We have all the information here. So I know, I'm in China this week. There's a Chinese company in silico here who just, published on Nature Medicine about the first kind of AI-driven data, drug that's now in phase 2 clinical trials. So, really a lot of exciting things are happening around us. So fantastic discussion. Thank you for sharing your experiences and expertise with us today. I think we've all learned a lot, and it's really an hour well spent. And we really appreciate everybody here, the panelists and the audience for really, utilizing. I know everybody's very busy these days, the time with us here. So maybe with that, I will bring Melanie back to conclude our session.
Yeah, thank you everyone again today for your time and your important research. We would also like to thank LabRoots and our sponsor Olink Proteomics for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us as well and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care and goodbye, everyone.